Thanks everyone for being here and hello to all those joining us virtually. It's my privilege to introduce today's speaker, Professor Anne Simon. Um, Dr. Simon has been a professor for a long time, full professor at UMass and then at Maryland for what, like 20 years or something? 24 years now. Um, in addition, I suppose to the lay audience, she is most known for her work on the X-Files. But um, for those of us in the virology community, so if I can editorialize a little bit, a plant virologist has to work twice as hard and be twice as smart as an animal virologist, or 4 times as hard as an HIV virologist. That means the stuff that she's doing, is going to be really good. And I first saw her work when I was 18, 19 years old at ASV. I was very impressed with the beautiful characterization of the relationship between nucleic acid sequence and the function of nucleic acids. And that's a topic that I think even for most of you non-infectious disease people in the room, it's certainly something very near and dear to us at the Protein Data Bank and at the Institute for Quantitative Biomedicine. Some of Dr. Simon's other elements of her CV. She consults for the feds on biosafety, biosecurity, and in addition to government and academia, also has this industrial experience as the former CEO and now chief scientist of Silvec Biologics, which spins out of some of the research that she's done. There's a lot more I can say with a CV miles long, but rather than that, I think I would like to turn it over to see what you have to tell us about today. So thank you very much for being here, and I'm very much looking forward to your talk.
Well, thank you for that really nice introduction. I was just reminiscing about the last time I was here. I was really sick, but I'm not anymore. So it's great to be back here at Rutgers, and I'm going to tell you a story today about these really interesting. And do I have a pointer? Sir? Or I mean, I have a pointer, like I have a finger. But finger works. Maybe there's a pointer. Okay, well, we'll get a pointer. But these really interesting viruses that have only recently come to light. And there are reasons why they've just been known in the past, let's say, decade, because they're virtually symptomless. And so it's taken these kind of metagenomic studies where you're literally taking a square meter and saying, what are all the viruses in this square meter? And all of a sudden they're finding these. So the talk today is on these novel features of these umbra-like viruses. And I think that, can I advance with, okay, so I can advance. Hang on, it's going the wrong way. Okay, so I've divided the talk into 3 sections. So I'm going to talk about the unique properties of umbra-like viruses. Then we're going to talk about their transcriptomes. We're finding out what's going on inside plants with these plant viruses and getting really astonishing results. And then I'm going to talk briefly about -1 frame shifting. So to introduce this talk, I want to start with what are the minimal coding requirements for an infectious plus-strand RNA virus. And, firstly, of course, you have the subunits of the RNA-dependent RNA polymerase. You have to be able to replicate. A coprotein, obviously, you need to encapsulate your genome, and then vectors can pick it up and transmit it to other plants. You need to be able to suppress the plant innate defenses like RNA silencing, and you have to be able to suppress the salicylic acid signaling pathway. Most importantly, you need movement proteins. And so movement proteins, plant virus movement proteins, are considered the key signature feature of plant viruses. So if you have plant viruses that are related to animal viruses, like the rhabdoviruses, you have really good synteny going across the genomes, but the plant viruses will always have an additional open reading frame, and that's a movement protein so that they're able to move through plants. So here's a typical plant virus. It's a virus I used to study called turnip crinkle virus. And so here you've got the replication proteins right here. And this is very typical of viruses that don't have and encode their genome using polyproteins. But you have this first open reading frame, and then either a read through, which is right here, or a frame shift that then allows you to make the RNA-dependent RNA polymerase. You then have movement proteins here, 1 or 2 usually, and then your capsid protein. And since plant viral proteins are multifunctional, many of them, the coat protein is also, for turnip crinkle virus, is also the silencing suppressor. So today's talk is going to involve quite a bit about movement proteins, at least this first part. So I want to start by talking a little bit about them. So movement proteins are always encoded by plant viruses. And the reason for it is that plant viruses have to be able to move through these connections between plant cells, these called plasmodesmata. And so the movement proteins need to enlarge the plasmodesmata, apparently. And then the movement proteins have to be able to traffic the RNA through the plasmodesmata. And in plants, movement is usually as a ribonuclear particle, and it can also be virions like animal viruses, but it's mostly movement is RNA. And for movement through plants, you have what's called cell-to-cell movement. That's through mesophyll tissue. But when you enter the veins for this long-distance movement, the plasmodesmata change. The plant virus has to be able to get through into the vascular system and then back out. There are also plant viruses that spend their entire life only in the vascular system. So these are just some examples of movement proteins. There are 4 different types. There are these very small hydrophobic proteins found in caromoviruses like turnip crinkle. There's what's called the triple gene block, which are found in Potex viruses and a number of other viruses. Poteviruses are very strange. And they have their own unique way of moving, maybe with vesicles. It still really isn't figured out, but they have these 3 proteins involved in movement. Actually, I think it's 1 and then 1 and a read through or a frame shift right here. And then there's this protein right here, which is called the 30K protein. This is the most common movement protein, and it's called the 30K because in TMV it's 30 kilodaltons size. So if you look at what viruses have 30K movement proteins, you're not meant to actually see this right here, but it's a lot of different viruses. It's double-stranded DNA viruses. I think you have single-stranded DNA viruses. You have minus-strand viruses somewhere. You have plus-strand viruses, and they all use this, some version of this 30K protein in order to move around through the plant. So what do movement proteins have to do? They have to bind, it turns out they bind non-specifically to nucleic acids, which is a little strange because they very specifically move the viral RNA and not other RNAs in the plant, but they are non-specific RNA binding proteins. They increase the size exclusion limit of the plasmodesmata. As I said, viruses are moving as these RNA complexes or as virions, and movement proteins are targeted to plasmodesmata, and they're able to mediate their own cell-to-cell movement and the cell-to-cell movement of the RNA that they're bound to. So there are exceptions to these minimal requirements, but the exceptions are mainly viruses that don't encode coproteins. So an example of a virus that doesn't encode a coprotein is an umbravirus. So because we're going to be talking about umbra-like viruses, you need to know a little bit about umbraviruses, which we used to work on quite a bit. So umbraviruses have, of course, these replication proteins, They have two movement proteins. They have a 30K type movement protein here and a second movement protein that's involved in long distance movement. So this is their cell-to-cell movement protein, but there's no capsid protein and there's no silencing suppressor. So viruses that don't encode co-proteins, they're always associated with a helper virus. So here's the helper virus for umbravirus PEMV2. It's a virus called an enamel virus. And the helper viruses for umbraviruses, they're either enamel viruses or viruses or polariviruses. They're very closely related viruses. And there's always just one. So what does the enamel virus get out of this association with umbraviruses? Because in the field, you only find umbraviruses associated with a helper virus. So the umbravirus actually provides the cell-to-cell movement ability to the enamel viruses, which is normally restricted just to the vascular tissue. In return, the enamel virus provides a silencing suppressor and also provides the co-protein. So it's this very nice relationship between umbraviruses and their helper viruses. So here now are the umbra-like viruses. And this is really the first time that they've been kind of grouped. I've been finding these in the database, finding them in papers that get published where they're saying there's something here, we don't know what it is, and it turns out to be one of these. But I've been dividing them up. So I've divided them into two groups. There's the group 1, which is everything that is not a group 2. So here's, it made sense to me, but it actually makes some scientific sense as well, because if you look at the phylogenetic tree, here's umbraviruses, here is everything except group 2. So they kind of all fall in the same place. And then here's the group 2s, and the group 2s are divided into 3 different classes. So there's these called class 2, and you have dicot infecting ones, dicot, and monocot infecting ones like grasses. And there's a clear difference between them. They all have this additional protein here, but the monocot ones have an embedded open reading frame that are kind of different from each other. This one down here, it's probably not even making this truncated protein, but it's a taro apparently is a monocot. I had to look that up. So the interesting thing about these, let me go back here, is these additional open reading frames that they have. So you have ones that, this one is the most closely related to the umbraviruses, and it has a 30K movement protein. Okay, great. What these are doing, we don't know. So all the ones that are in light green here, we really have no idea what they're doing. And then we have this one we call open reading frame 5 here that I'm going to be talking about today. The obvious thing when we started looking at open reading frame 5 was that it was a movement protein, because all plant viruses have movement proteins. And so, you know, just like umbraviruses with their additional movement proteins, we figured that would also be a movement protein. Okay, so another interesting thing about these is that some members, like the class 1s, are just a polymerase. I mean, that's all they are. And this one right here has lost its ability to make this extra open reading frame. So this is very curious. I mean, are these real viruses? Are they found by themselves? Are they found with helper viruses? Well, it turns out that only 2 of these are known to be associated with a helper virus. Umbraviruses, every single one. And this one, that looks just like an umbravirus. At least it has this 30K type movement protein. It doesn't have the embedded protein, but it's similar. So these have helper viruses. And then it turns out that all of these do not. So these are the ones that are known to not be associated with a helper virus. In other words, they have literally been found in plants all by themselves, as far as the data reports. And that includes some pretty odd ones, like these right here, that are just a polymerase. And it includes this one that's just a polymerase. And then all of these, and probably as the years go on, probably all of these are going to end up with red stars as being found all by themselves. So how is that possible? That they're found in the absence of a helper virus is really the first part of this talk. So in my lab, we've been studying mainly these two. We're trying to study a couple of the others. They're a little bit more difficult. We're trying to study this one and this one right here, running into some problems. But these two are pretty easy to study. So they're very closely related. They're 90% the same. This one was found in citrus, but it was only found once. It was found in like 1960 in 4 limequat trees, lime cross kumquat, limequat tree. Yeah, I mean, like, who eats limequats? But okay. In 4 limequat trees, it had some interesting little symptoms on the leaves, just some pretty yellow lines. And so it was sent to UC Riverside to find out what was in their trees. They wanted to know. And the guy who was there, Weathers, was the citrus expert, did a lot of work trying to figure out what this is. And he said, well, he didn't think it was a virus, and he didn't think it was, it wasn't a fungus, wasn't a bacterium. So he couldn't identify what it was. I think he got like 2 Nature papers for that. We don't know what it is. Yes, let's publish that in Nature. I guess it was a little easier back then, maybe. Oh, I think he had a PNAS paper too on, we really don't know what this is. But it took many years later, it was like 2011, when the person who replaced Weathers at UC Riverside decided to do RNA-seq of the trees that were left over from Weathers. And this was in all the trees. And it was all by itself in some of the trees. There was no obvious helper virus. So this was very odd. And I figured they did it wrong. We asked to see the data. It's like, yeah, of course there's a helper virus, you missed it. And we couldn't find it. So this seemed to be all by itself. So one of the first things that I did when I started studying it was determine its secondary structure. This is not a trivial thing to do. It isn't just put it in M-fold and press a button. Okay, it doesn't give you anything like this. But it was my COVID project. I was stuck at home. I thought, okay, let's figure out the structure. So I figured out the structure. And to figure out the structure, it really took a combination of shape RNA structure mapping, a little bit of M-fold, and a lot of phylogenetic comparisons. It turns out that all of these class 2s have genome structures that are very similar, even though some of them are only about 45% the same as far as at the nucleotide sequence level. And so we made infectious clones of these two. And here's CY1 right here. It does not make a subgenomic RNA. There's no reason to. Here's CY2. It does make a subgenomic RNA. Right here, this is the subgenomic RNA promoter. We know where the start site is based on a lot of work that's been done on similar viruses. It was very easy to predict. And one thing that I just want to point out, because it's going to be important later, is look at the smear here. See the smear? There's smears, and you frequently get smears. I mean, when is a northern perfect, right? You get these smears, and so what do you normally do when you see smears like this? You ignore it. But it probably shouldn't have been ignored. So we'll talk more about that in a little bit. So we have the structures of these viruses, and the structures turn out to be really important. Now, we wanted to know whether or not these were able to move systemically. So we got infection in protoplast, but those are just single cells. And it was really astonishing that when we delivered it into plants by agroinfiltration, a very common technique, we found that it goes systemic. And it turned out that the graduate student who was doing this, Brianna Liu, one of her first 10 plants, it went systemic. And if that one plant had not gone systemic, none of this work would have been done. There would be no company, and I'd probably be having trouble getting grants. So because one of the plants gave symptoms that looked like this, it was pretty obvious that something was going on. So the symptoms that we get with either one of these, this one infects quicker than this one does. But you start out with this kind of heart-shaped leaf, right at around 3 weeks for this one, 2 weeks for this one. And then the plant literally doesn't grow anymore. And you get little leaves all over. It's really quite astonishing. I mean, I've never seen a virus do anything like this. And you get nodules on the roots as well. Just these they look like galls actually on the roots. Something that we found recently that I just wanted to present here because there'll be some plant people and I thought maybe you'd know what might be doing this is what we get at 22 degrees. Okay, so we went a little bit further, but I'm just talking about this right now. This is what we get at 22 degrees, these symptoms. At 33 degrees, actually, we get the same amount of virus. So that's kind of interesting. Usually when you increase the temperature up that high, virus levels certainly go down. The virus has inhibited the infection. But what we get are these lateral shoots. And we've never seen lateral shoots coming off of Nicotiana benthamiana like this, but they're on every single plant. So if someone has a good reason for why that should happen at a high temperature, I'd be very interested to know. So we did fluorescence in situ hybridization of both of these, and we did it also of CY2 in the absence of open reading frame 5 when we made mutations to get rid of it. And what we found is that they're phloem limited. We found it only in the vascular tissue, and there is quite a lot in the vascular tissue. So this is in what are called sieve elements right here. They're in the cells associated with sieve elements, companion cells, and phloem parenchyma cells. And there seemed to be a lot in roots as well. So they were in roots and in petioles, but phloem limited. Okay, but even phloem limited was a big surprise because it's just a polymerase right here. So this was quite surprising. So we wanted to ask the question, is open reading frame 5 a movement protein? And since all plant viruses have movement proteins, and a paper which is going to be published hopefully soon in PLOS Biology, which deals with this, we actually got rejected a year ago, a little over a year ago, from PLOS Pathogens, because one of the reviewers said, it's absolutely impossible that this is moving. All of this work is wrong. We didn't get that this time because of all the work I'm about to show you. We're very surprised that this is going systemic in plants. It really shouldn't be able to, but we figured this was a movement protein and that this is somehow moving in the absence of it. So we did a lot of work. This is a summary of a lot of work. It has no effect on replication, this protein. It has no effect on tropism. It's still vascular limited. The symptoms appear 1 week earlier when you're encoding open reading frame 5. And it supports systemic movement, definitely, but it's not required for systemic movement. And movement proteins are required for systemic movement. So we took a look at it. We bow down to AlphaFold all the time because it's such a wonderful gift to science to be able to actually look at these proteins here. So this is a typical 30K movement protein, the one found in this umbravirus right here. So you see this jelly roll, this single jelly roll right here, this alpha helix right down here, these N-terminal and C-terminal extensions. Very typical 30K protein. And here's what you get when you fold up open reading frame 5. So initially we looked at this and said, it's a movement protein, right? I mean, it's got this jelly roll that looks pretty similar. It's got this N-terminal extension, but it kind of looked like it. And then the following paper came out. So this is a paper that came out in PLOS Biology from, Valerian Dolia and Eugene Koonin are probably known by any of the virologists in the room right here, and Mark Kupiuk. And what they came up with was a hypothesis that said that the 30K movement proteins are actually evolving or evolved from these single jelly roll coproteins. And that coproteins, there's a number of coproteins with this single jelly roll, very similar to what you find in movement proteins. And if you think of what the progenitor of a plant virus was, well, it was probably an insect virus or a nematode virus or something like that. It was a virus that had an RDRP and a coprotein. It had to have those two things. And then in the paper, they say, well, maybe the coprotein open reading frame duplicated. One of them was able to evolve independently into a movement protein. And then you got horizontal spread among many viruses. So this is their idea for evolution of these 30K proteins. So So all of a sudden we're stuck. Is this a movement protein or is it a coat protein? And so we made this little list. These are properties of 30K movement proteins. Here are properties of single jelly roll coat proteins. So 30K movement proteins are essential for movement of the virus. The coat proteins can support movement. Okay, well, that's what we were finding, that it can support movement. A little hard to do. There we go. One of the key signature features of 30K movement proteins is a particular aspartic acid residue right after one of the beta sheets. So it's in a very obvious place. You can line these 30K proteins up because of it, right here. Nice aspartic acid here. It's also found in some coproteins. I don't know if you can see down here, but these are a couple of coproteins you can find it in. But there's a lot of coproteins that don't have that aspartic acid. This is the open reading frame 5. None of them have an aspartic acid in that location. So another check mark over here. So the third one is in 30K movement proteins. They have an even-ish plus charge distribution across the protein. In the single jelly roll coproteins, the N-terminal region is more positively charged. It's probably what binds the RNA. So what do we see here? Here's a movement protein right here. I don't know if you can see this, but if you look at your positively charged residues, it's going up and down, and there's nothing unique about the N-terminus. If you look at this coprotein right here, the N-terminus is very positively charged. If you look at open reading frame 5, it's identical to this coprotein. So another check mark on this side. So what about additional domains? 30K proteins are known to have additional N and C terminal domains. These can have additional domains. Open reading frame 5 has only an N-terminal domain, additional domain that's unstructured. And it does not have a C-terminal domain. So again, that puts it over here. Then in the paper, they reference this long alpha helix right here in between the two beta, the collection of beta sheets right here. And open reading frame 5 did not have that. So it did not contain that alpha helix. Okay, so then, sorry. Okay, so then there's the last one. Coproteins generate virions. So at this point, after finding all of this, we started to change our minds that open reading frame 5 is probably not a movement protein at all, that it's a coat protein. Now it makes a lot of sense. I mean, I don't even know why we were confused for a year, but we wanted to know whether or not it generated virions. So what we did was a very, very mild prep, very, very simple virion prep, because obviously we don't know anything about this. We didn't know if we'd find anything. So we have a 70% sucrose cushion, 25% sucrose cushion in between. And what we found is that with the 70% sucrose cushion where you can find virions, we did see something very high molecular weight right here in CY2, but we also, kind of saw it in CY1 as well. That didn't tell us much. But then we found in this fraction right here, in this 70% sucrose fraction, we found open reading frame 5 protein. So it was in that fraction, okay? Then we did a northern blot to ask, is the RNA in this fraction? And yes, the RNA was in the fraction. Both the subgenomic RNA and the genomic RNA was in this fraction. So then we went ahead and did some electron microscopy. Go back. There we go. So we did some electron microscopy, and what we saw was just really astonishing. So here's just uninfected Kochiana benthamiana. There's nothing in the sucrose cushion. CY1, there's these kind of amorphous stuff going on here. So there's certainly something going on in these plants that are infected with CY1, which doesn't make open reading frame 5. But in the plants that were infected with CY2 that make open reading frame 5, we saw lots of these little particles that look very much like a virus-like particle. They're only 14 nanometers, so they're really small, but they're there. And occasionally we see these two sizes as well. So there's this size right here, which really looks icosahedral in shape. And then there's this one right here. So, you know, at the present time, all we know is that in these plants, we see what look like virus-like particles. And obviously there's a lot more work that needs to be done there. So it looks like it's maybe generating virions. And at this point, we have concluded that open reading frame 5 codes for a coprotein. If you look at coproteins that are single jelly roll coproteins like this one, you can see the similarity right here. They both have N-terminal domains that are actually nucleolar localization sequences. And both of these do go to the nucleolus, and it's very easy to detect them in the nucleolus. I don't know what they're doing in the nucleolus. They're probably picking something up there, maybe fibrillarin, but they both segregate to the nucleolus in infected cells. So it looks like what we have is a coprotein. And if you do a phylogenetic tree of just that open reading frame, so here's open reading frame 5 right here, the most closely related proteins are these coproteins. The coproteins from barley yellow dwarf virus, from this polarivirus, the sabimovirus right here. So interestingly enough, the coproteins that are the most closely related are coproteins of umbravirus helper viruses. It's those coproteins, which maybe they picked up when they had need of a helper virus. I don't know. So what this means is that these group 2 umbra-like viruses are unique. These are the very first plant viruses that have been found that do not encode a movement protein. They apparently encode a coprotein, except for CY1, which explains their lack of a helper virus. You don't need a helper virus if you're able to make virions and be picked up by vectors and transmitted to other plants. But you still have the problem. You have to be able to move in the host, which means they must use a host protein for movement, like the original plant viruses. So the question is, what's the host protein that they're using for movement? So we looked. So this is a Northwestern using sap from cucumber. So CY1, which is the citrus virus, also infects cucumber. And it's very easy to get sap out of cucumber. So we looked at CY1. We also looked at a defective RNA derived from CY1 that I'll talk about a little bit more later. And then as a control or other RNA, we use the PEMV2. Okay, so here's sap from uninfected cucumber right here, and we radio label these RNAs and just see what they bind to on this gel, and they all bind the same protein. They bind this around 26 kilodalton protein right here. It's this one, this very strong band, and that strong band is a protein called PP2, phloem protein 2. This is the most prevalent protein in the phloem of higher plants. It's a very interesting protein. It's a non-specific RNA binding protein, but it actually is binding specifically to CY1 and the defective RNA compared with PEMV2. So maybe it's been called non-specific because they haven't found the specific RNAs that it binds to. It's the most prevalent protein in the phloem. It turns out this is the movement protein for viroids. So viroids are these small, single-stranded, circular infectious RNAs. They don't code for anything. They have to use a movement protein from the host, and the host protein that they happen to use is PP2. The function of PP2 when it's not moving viroids or other, infectious RNAs is not known. So one of the things that we did is we also did these EMSs to see whether or not binding to PP2 or whatever it was binding to, maybe PP2 and other things, whether it was protected against RNAs. So we added different amounts of SAP to either CY1, the defective RNA, or PEMV2, and all of them had this very sharp transition to this high molecular weight complex. This one, the transition was earlier, the one that binds best. But we also got the transition with PEMV2. However, we then treated with RNase. So this is untreated and treated with RNase. And what we found, if you look at PEMV2 here, is that if you add RNase, there's no protection. It's just the RNA just gets degraded. But if you look up here for this defective RNA, it's almost completely protected from RNase treatment. And up here with CY1, it is partially protected from RNase treatment. So there's clearly something different about the interaction that it has with CY1 and the defective RNA and PEMV2. We then went ahead and did assays where we pulled out CY1 from infected plants and found that it was bound to the same size protein, which using our anti-PP2 antibody turns out to be PP2. So what this is saying is that in vivo, it is complexed with PP2 in the phloem. Interestingly, also, if you take uninfected, this is Nicotiana benthamiana sap, but we could also do cucumber. The PP2, once you concentrate PP2 down, it polymerizes. So you see all this stringy PP2 when you isolate sap. If you take that exact same sap and add CY1 to it, all of the stringy PP2 disappears, and now you see these little like vesicles, sticky vesicles, and that's all you see. You don't see any of this anymore. So there's clearly something going on when you add CY1 to sap that is involving PP2. Oh, this is just, I want to show that. Okay, here's the structure. This is the cryo-EM structure of PP2. Notice the similarity with movement protein. So it's also a single jelly roll protein, and it's known to move into the phloem. It's known to be an RNA binding protein. We really believe that this is the host movement protein. So we did one more experiment. So we've worked very hard over the past few years to convert CY1 into a VIGS vector, a virus that can do virus-induced gene silencing. One of the things that we had to solve was stability, and I'm not going to talk about stability now. That will have to be at another date. But VIGS vectors are never stable. Animal vectors where you're adding hairpins to, plant vectors where you're adding hairpins to, they're not stable. They get lost almost immediately. They're gone. So one of the things that we did is we solved the stability problem. We can actually keep these viruses with inserts now for as long as we've done it, which is over 2 years in citrus. They're completely stable. So how did we do this? I can just say that all of the structure of this virus has evolved. It's very important. It has evolved to be exactly what it is. The structure of these hairpins is extremely important. And it's extremely important what the positional entropy is of every single nucleotide. Once you realize that, you can design hairpins that are mimicking viral hairpins, and those are stable. So if you're just making any hairpin you want, just because you want this sequence in there, so you make a hairpin and you put it in a virus, it'll be gone very quickly. But if you put in a hairpin that the virus thinks has evolved with it, it's very stable. Okay, so we made VIGS vectors with CY1 containing 2 siRNAs. One targets one PP2, another one targets a different, it's actually a family, targets another phloem-specific PP2. And what we found was the following. We found that our VIGS vector that's targeting PP2, if you're looking at relative PP2 levels here, here's a random hairpin. So if you just look, we're about 50%, maybe a little bit more, PP2 levels have gone down in these plants from this vector. If you look at the level of CY1 now, that's actually in these plants, There are plants where you can't detect it anymore, plants where the detection is much, much less. But there's definitely a significant difference between the one with a random hairpin and the one with a hairpin that's targeting PP2. So if you target PP2 such that the level of PP2 has gone down, it's no longer available to act as a movement protein, and the virus has a really hard time moving around in this plant. So based on all of this information, we've really concluded right now that host RNA movement protein PP2 appears to be the CY1 movement protein. And it's probably a protein like this that allowed the original plant viruses to be able to infect plants before they developed their own movement proteins. Why these particular viruses continue to use this right now, we don't know. Okay, so I now want to talk about this work that's very recent. It's not published yet, but we're very close to being able to submit this, analyzing a plant virus transcriptome. So why would you want to analyze a plant virus transcriptome? Everybody knows what's in a infected plant, right? I mean, you have full-length plus strands, you're going to get full-length minus strands. People that use double-stranded RNA antibody find that they can detect this, a double-stranded RNA, what they'll call an intermediate in replication, because you get a strong signal when you use double-stranded RNA antibody. So they say this is double-stranded. Then you would make full-length, a plus-strand progeny. You'll make subgenomic RNAs if your virus makes subgenomic RNAs. And then frequently you can get defective RNAs where you've got some 5' sequence, some 3' sequence, and you're missing something in between. So why bother to analyze a transcriptome when everybody knows this is what's in a plant? Well, we had some questions. I think this went a little quickly. So the questions were, does it differ during the infection in newly infected plants after long term? And is it different in different parts of the plant? I mean, do you see differences? So that was pretty much all that we kind of were expecting to see. What we use is nanopore sequencing, and it's very important that nanopore sequencing was developed because you really couldn't do this work without it. Nanopore sequencing allows you to sequence full-length RNAs starting at the 3' end. These are small viruses. They're about 3 kb or smaller. It's very easy to sequence full-length RNAs that are 3 kb using this technique. It stops about 13 nucleotides short of the end, and it's not very accurate. Okay, it's about 90% accurate. So this is not a good thing to follow virus evolution in a plant because it makes a lot of mistakes. It is said that the mistakes are random mistakes. It turns out the mistakes are not random. It makes very specific non-random mistakes. And just in case you're interested, because we're also going to be submitting that paper when we realize that was going on, it has to do with the structure of the RNA after it's fed through this membrane. So you make these RNA DNA duplexes, and then the RNA feeds through. If the RNA folds up right afterwards, it affects what's going on here, the detection of the bases here, and that's where you make mistakes. So it's very easy. It was not difficult to figure out that these mistakes were not random. You can actually use it to map strong structures too. Okay, so what would you expect to get from full-length in vitro transcripts? If you're good at making full-length in vitro transcripts, this is what you should get in your coverage map. So you're looking at every read that contains this base or 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 this base. And so full-length transcripts, if you've been, if they're not degraded, this is what you would expect with every single one of these being full length. What you actually get is this. So you get something very similar. And all of these are right here are full length. Okay, so if you're really seeing in a plant that most of the RNA that you're seeing is full length plus strand, this is what you would expect to see. Full length plus plus full length minus. This is what you'd expect to see. So what do you actually see? So this is 2 weeks post-infection of Nicotiana benthamiana, and I'm showing the results for roots and leaves. So The first thing that you notice is it doesn't look a thing like this. You have some, what look like, RNAs, like long non-coding RNAs going on here. You've got these little grooves in here. Roots look a little bit more like this, and roots don't look like leaves. So here's what you get after 6 weeks. Now you're really seeing something strong here. You see the same grooves in here. The roots are a lot more what we thought was just, full-length RNA here, and you don't see the same thing. You don't see the grooves. You don't really see these very prominent, what we thought were additional RNAs. It turns out these are additional RNAs right here. So we looked in leaves at 14 weeks post-infection. This is with CY2, because we wanted to see if we could detect the subgenomic RNA. And sure enough, here's the subgenomic RNA right here. It was exactly where we figured. The start site was exactly where we predicted. But it looks pretty much like this. So then it turned out we got a sample from a field of CY2. We got an infected hemp sample. It is very difficult to get growers to give you a sample of hemp from their field. They will not give you the roots. Okay, so we can't get the roots, but we got some dried leaves. Okay, we were happy. And this is what we got. This is the coverage map in hemp after a long time, probably. What you're seeing here is a defective RNA. Probably 95% of this is this one defective RNA right here. And down here somewhere are your full-length plus strands. This is also the defective RNA right here. So this drop off right here, and then this drop off right here is also a very specific defective RNA. It's slightly different from this one. It's got the same junction here and a slightly different junction there. Okay, so more about this transcriptome. We see two long non-coding RNAs that are 5' proximal. We see this 281 fragment and this 671 fragment. It turns out that this 671 fragment is very good at reducing host defenses. So maybe that's why it doesn't need a silencing suppressor. We also see this drop off right here. We knew that this was our defective RNA. If you look on our structure, so nice to have a structure, here's where the 281 fragment, it goes from the end to 281 right here. It's this RNA. The 671 is right at the base of this, we call this the bridge. So the polymerase has gotten to here on the minus strand, it's fallen off, and it picks up right here. So it's really very, very close where the other site is at 2420 and goes to the end. We also have defective RNAs that just cross here. So it ends here and starts right there as well. The grooves turn out to be really interesting. These are actual endonucleolytic, we believe, cleavages in the RNA at very specific places, and that's why you see these as grooves. And they map to these single-stranded regions, virtually all of them do. I should mention that the colors right here are for inline probing. So one of the things that we wanted to know is inline probing is where if you have a flexible residue in the backbone of your RNA, it's able to cleave the backbone. So it's a way of also looking at structure, just like shape, but different. And we thought since inline cleavage produces fragments, we could use inline cleavage associated with nanopore sequencing to try to see if we could use that to map the structure. An awful lot of this looks good. Some of it is weird, but an awful lot of this, the stronger colors map to the single-stranded regions. So it worked to an extent that way. So we have these grooves. We also wanted to know exactly how much of these RNAs are in here. How much of this down here is full length? And originally we thought that like down here, we're dealing with full length genomic RNA. And it turns out we were completely wrong about this, completely wrong. How much full length genomic RNA is there in this 6-week sample? If you consider only the CY1 reads that have CY1 in them, There's 3.2%. 3.2% of the reads are full length. That's it. The rest of this is all fragments. It's the smear in the gel. You get a lot of fragmented virus, a huge amount, maybe 80% is fragmented virus. The 281 fragment here is 14% of the CY1 RNA in the leaves is this one long non-coding RNA. You don't find it in roots. Full-length genomic RNA is about 5 times more prevalent in roots. And we think now that roots are a repository for full-length virus. And that would make sense. I mean, if you look outside right now, you have all that pretty snow on the ground. I can say it's pretty because we didn't get snow in Maryland, so it's only pretty here. But there's no leaves, right? Except the plants where the leaves don't die off. So if you've got a virus infecting there and your leaves are all dying off, where is the virus going? There's all this virus in the roots so that when the leaves come back and you start getting flushes in the spring, the virus can emerge from the roots and take over. I mean, so it makes sense that roots are a repository. Okay. The 671 fragment all by itself is about 3% of the RNAs. The defective RNA is about the same. There's about 3% defective RNAs here. So this was very unexpected. This amount of full-length virus in the leaves was very unexpected. And then, of course, we thought about this. Here we're seeing it's almost all defective RNA, and probably there's about 3% full-length virus in the leaves here. So it isn't something associated with Benthamiana. It looks like it's something which is much more common. And I want to point out one thing, which I think is really important. If you're doing RNA-seq to look at what a virus is in a plant, okay, what exactly are you sequencing at the 5' and 3' ends? Are you really sequencing your genomic RNA? Or are you sequencing something very different? And my guess is you're sequencing defective RNAs, you're sequencing, I don't know, fragments. There's an awful lot of stuff you're sequencing that is not your full-length virus here. And that's probably why when you try to make infectious clones from sequences in GenBank, you get things that don't infect. So, RNA-seq can discover viruses, but I don't think it can give you accurate sequences of those viruses. Okay, so what else is in this transcriptome? Well, you get these. These are called plus-minus foldbacks, and we've known about those for a long time. I actually have a paper from like 1970, no, not that far, 1997 on foldbacks. Okay, so I've known about this for a long time. So foldbacks are generated by RDRP when they fall off the template. So you've got an RDRP that's making a plus strand, let's say, and if it falls off at position 671, it can either make a defective RNA, or it can just fall off and release the nascent strand and give you this fragment, or it can do this foldback thing, where it literally uses the plus strand it just made as a template and makes a complementary strand. It turns out that 38% of all of the minus strand reads are in foldbacks. That's a lot of foldbacks. And if you think about it for a second, this is going to react with your double-stranded RNA antibodies. And it is not an intermediate in replication. It is a dead-end product, which may or may not have a function. I mean, finding this much, it's known to have a function for minus strand viruses. It's not known what's going on with plus strands, but my guess is this is having a function right here. So we looked at exactly where the junctions were between the plus strand and the minus strand complement. And then you usually get some non-templated bases here or other things. So it turns out we have 2 peaks. One is at 671 and one is the full-length virus. So what that's saying is that the 671 fragment is made by the RDRP, which made sense because it's half of a defective RNA. It made sense that this was RDRP derived. But there are no foldbacks for the 281 fragment. There's 0. So we actually think that this may be derived in a different way, maybe exonuclease derived, and we're studying that. The last thing is we were quite surprised to find that an awful lot of the RNAs are polyadenylated. There is no poly A tail at the end of these RNAs. There's no template. However, they are polyadenylated. And it turns out you can use oligo-DT primers, and you can amplify up just as much as you can by using random primers. The amount of poly-A's that are present on the different, the genomic RNA versus the fragment, the 281 fragment versus the 671 fragment are different. There's less on the genomic RNA and more on these fragments. And so it was very interesting how we found this, because one of the steps you have to take in nanopore sequencing is you polyadenylate your RNA. So of course you're going to have poly-A's. You polyadenylate it. But we had way too many RNAs. We had way too many A's on there. It was like, why are there so many A's? So we looked at RNAs that definitely are not polyadenylated, like ribosomal RNA, and here's what you get. We looked at a few different ones. You get this short peak when you do nanopore sequencing. And then you look at CY1 and you see that you have a lot more A's on the end. There's about 100 extra A's on these viruses. We don't know what it's doing. So we'd be very interested in knowing, is it involved in translation? I don't know. But it was very unexpected to find that these RNAs were polyadenylated. Okay, so very, very quickly, because I'm running out of time, I'm just going to tell you very briefly about this really exciting result we got when we were studying -1 ribosomal frame shifting. So for those of you who don't know a lot about frame shifting, although I don't know how that can be with Nilgün tumor here, who knows a lot about frame shifting, so hopefully any students in the classes will have that, and hopefully others, this is just how an awful lot of plant and animal viruses like SARS-CoV-2 make their polymerase. So you have a frame shift here or a read through. A frame shift basically has a slippery site, and it has some sort of impediment for the ribosome. So the idea behind frame shifting is the ribosome hits an impediment, shifts back one base, and then continues. So instead of terminating translation, it stops, shifts back, continues. Now, one thing that they have never explained in any paper I had ever seen, maybe I'm wrong, but I haven't, is if this thing stopped the ribosome, how do you get rid of it? How do you get the ribosome to keep going? Somehow nobody sort of talked about that. And we think we found an answer for CY1. So another thing is, how do you study frame shifting? Frame shifting is almost always studied using these dual luciferase constructs. So what you do is you put your frame shifting site in the center here between these two luciferases. This one is 5' terminal. You get luciferase made. If you get the frame shift, you make the second one so that you can see the ratio between the frame shifted product and this product, and it gives you your rate of frame shifting. Virtually everyone studies frame shifting like that. And you're going to see why that's been a really bad idea, because it's probably given an enormous amount of incorrect information. Went one slide too far. Trying to go back. Okay, so here's the recoding site on umbraviruses that we did this like 10 years ago. There's a pseudoknot here. This sequence right here does a long distance interaction with the 3' end. That's very common with viruses in the Thomas viridae, is that if you have a read through or frame shift, you have a long distance interaction. So automatically that should get you thinking, well, if all you do is take this out of context and put it between the two luciferases, you don't get the 3' end here, which is critical for frame shifting, by the way. Okay, so you would get a much lower rate of frame shifting without this. This is the frame shifting region in CY1. You get this hairpin. You always get this hairpin right before the frame shift site. Here's our slippery site, and here our stop codon. And I want you to just notice the colors here. This is a shape. The shape data is on here. And if you see colors, that's very shape reactive, especially the purple. And what you see here is that this structure does not fit the shape data at all. And there's a reason for that, is that the shape data is actually reflecting an inactive structure. And this is an active structure. One of the problems with the shape and using computers to determine what the shape is telling you. So we wanted to study this, and I'm just going to really summarize what we got. So this is a summary of what we found. We found 6 tertiary interactions with this frame shifting site. We have this one here. It's this terminal loop with this 3' end. So here's the one that we generally find in viruses in the Tombus viridae. But there's also a second one right here. And when I'm showing these on CY1, it's 100% conserved, these interactions, 100%. So you have either this one, which is the active conformation, or you have this one, which is an inactive conformation. This does not stabilize this structure. It does the opposite, okay, in the full-length virus. We also have this very interesting one here. There's 4 bases here that do a very long tertiary interaction with sequence beyond the bridge. So there's another 1,800 bases here, and then there's this. So in space, it's very close, but it's 1,800 bases away. And then there's this local one as well, and they share one base. Because they share one base, it's an either or right here. Then there's these 2. These turn out to be really interesting. The blue one, which pairs here, and the red one in this asymmetric loop, it's always this asymmetric loop, that pairs on the other side here. And we'll talk about that one. So this is just showing they're 100% conserved in all these different class 2 viruses. I'm not going to go through here. I just want to point out that here's this really interesting one, this or this. It turns out that this one right here, the dark green, that's the really important one. That you can make single base changes, compensatory, it comes back. Here's the wild type, single base, compensatory, it comes back. This local one is not as important. It's right here, single and compensatory. The main one is this one right here. And the shared base, the data is very strange. You can change it to an AU, but you can't change it to anything else. So very, very important what those bases are. Here is the inactive structure that fits the shape data. And this is 100% conserved as well, where you have this now kissing loop interaction here. The blue sequence is now here, paired over here, and you no longer have the 2 green sequences, that long distance interaction over here. So here's the model that we have. The model is the ribosome goes until it gets to the stop codon in the A site. If you've got the inactive structure, it terminates translation. You make P21, and the ribosomal subunits come out. We get, if you look at how much frame shifting we get about 30% frame shifting here compared to the umbravirus. Then something has to happen. Some event has to happen that then picks up on this very, very important structure right here, this dark green structure. So what happens is you get a transition then over to your active structure, and then you go back one base. How do you get rid of this? That's the 2 other tertiary interactions here. First, you have the blue interaction that comes in, that unzips it. Then the red interaction comes in and unzips it. And now you have unzipped this structure, and that allows the ribosome to go through. So this, I don't know if other viruses do this, but RNAs unzipping the recoding structure is a way to get rid of it. So what I've tried to show you here is that if we had just done a dual luciferase assay, we would have missed every single one of those structures with the exception of the 2 local ones, one of which is in the inactive structure and one of which isn't really important. So it is very important to study these things in situ in the virus. And my last slide is summary. So most umbraviruses are unique in that they don't encode movement proteins, encode coproteins, require no helper virus. It looks like they're using host-encoded PP2 as a movement protein. We talked about the transcriptome, very surprising results, and ribosomal frame shifting, which should be studied in situ. And I thank the people from my lab and the funding sources. And I want to mention that all of the RNA structures are drawn using RNA Canvas. That's our program that we developed for visualizing RNA. It was the only way we could solve these structures, is using this program. I highly recommend it. It's free, easy, addictive, but really fun to use. Okay, thank you very much. Sorry, I went over.
So we have -7 minutes for questions, which I think means hostile comments.
Oh,
No,
sorry.
any questions from the audience?
I don't get the 5 stars, right?
All right.
Get the 1
That's
star.
it. Okay, let me start. Do you have a question? Okay, let's start with someone who won't meet with the speaker later.
Okay. Yeah, it's a really nice talk. I really enjoyed it. So my question is about the PP2 protein. It's amazing that you found the host protein that helps the movement of your virus. So does PP2 help other viruses, like without movement?
So that's a great question. I think this is the very first time that a host protein has been involved to be essential, really involved in movement, not some peripheral activity. I think that once this gets published, other people are going to search to see whether or not PP2 is also involved in helping the movement of their viruses as well. So I think we will hear about that as the years go on.
Okay, let's take Eric next.
Hi, Eric.
Hi, following up on that, I mean, great talk,
Oh,
of
thank
course.
you.
There's so many exciting things, but the one thing I was going to ask is, like, since you can now suppress PP2 so well, is there a phenotype in the plant?
Okay. So excellent question. PP2 is why citrus plants are dying of citrus greening. Okay. What happens in citrus greening is the bacteria infects the citrus, and PP2 levels go way up. It polymerizes. You get all that sticky, gunky stuff. It goes into the seed tubes, into the seed pores, and it blocks sugars, and the trees die. And we have about 10 more years before there will be no more citrus on this planet outside of greenhouses because of this bacteria. Obviously, we are trying to do something about this. So yes, we see a phenotype when we add the virus. Okay, so they did make GMO plants without PP2. Those plants are dead. They can't do that. If you sequester PP2, it looks like you also cause more movement of the bacteria. So it looks like that the plant is trying to sequester the bacteria by clogging up its sieve tubes and killing itself. So it does a bang up job of sequestering the bacteria and dying. So, so there has to be some, something we're going to have to do in addition. I think we have to clean out the PP2, but we've got to clean out the bacteria as well. And so maybe there's a solution in there, and the company is working on this.
So I regret that I think we don't have time for more
I'm
questions. Although you all should feel free to stick around after. I would like to present you with a little bit of swag from first from the protein data bank. So you, of course, you can look at some structures that are a little more rigid than some of your RNAs, and also our PDB. Yeti.
I love this. Yes, I
So,
love these.
thank you very much for visiting. Thank you all for attending, and we will now have the student lunch.